Hello friend, welcome to the channel Pharmacy Dictionary. In today's video, we are going to complete and summarize the topic of movement of drug across the cell membrane. And for this whiteboard session, we have to study about the facilitated diffusion. What is the meaning of this facilitated diffusion? Let me clear you the meaning of diffusion first, then I will come to the meaning of facilitated. Diffusion is a process of movement of molecule from the region of higher concentration toward the region of lower concentration without the need of energy. But in this case of facilitated diffusion, facilitated means to need support from any other molecule or any other substance to fulfill the process, to complete the process, to achieve a desirable response. That is called facilitated. So the diffusion is based on the help of other agent, that is considered as a facilitated diffusion. So what are the basic points, what are the basic consequence, and what are the basic difference of this facilitated diffusion from the other type of diffusion? So first point says this is a unique form of carrier transport. That means it is a form or type, or we can say subtype of carrier mediated transport. We have explained you the meaning of carrier mediated. That means something is happening with the help of some carrier which is being carried out by the particular carrier across the cell membrane. So that means facilitated diffusion is a type of that particular carrier mediated transport system which is different from the active transport. It is different from the active transport in the meaning of energy dependent. That means, as you can see, in the case of active transport there is a need of energy, and in this case of facilitated diffusion there is no need of energy. So we can say in one word that is it is energy independent, or it is not dependent on the energy, and that is a major difference between this facilitated and the active transport. Now coming to the second point, movement. What is the movement of the molecule of the drug? Where it is taking place, from which side to which side is happening. So in this case, movement of the drug molecule occurs in the direction of concentration gradient. What is the meaning of concentration gradient? That means the difference between two molecules on the basis of their concentration is simply known as the difference of concentration. I have two examples to explain you. Suppose in this plate, in this part, in this substance, the concentration is higher as compared to this one. So the movement of molecule will be take place from the higher concentration toward the lower concentration. That means there is a difference of concentration between these two substances. It may be anything, it may be a liquid, it may be solid, which is based on the process of diffusion. So coming to the next point, that is highly specific for substance, this carrier mediated, and we can say the type of carrier mediated, that is facilitated diffusion, is very specific for specific type of molecule. These are uptake of glucose by cell and uptake of vitamin B12 by the intestinal cell. In general case of example, this drug or this molecule are not taken by different types of or other type of diffusion process, but these are specifically allowed for the facilitated diffusion. In this case, a substance which carry this drug molecule will help to move that inside the cell, that is cytosol, and then they go back toward the surface of cell, so that they may help another molecule, another drug molecule, we can say. So these are the basic consequences of facilitated diffusion. In the next whiteboard section, I will explain you all this point with the help of respective diagram. Now coming to the endocytosis and exocytosis, the last transport mechanism for the cell. You can download the PDF of different cell membrane movement via the clicking link in description box. So what are the endocytosis and exocytosis? Endocytosis and exocytosis are the specific phenomena, or we can say a specialized phenomena of movement of molecule either inside the cell or outside the cell, because endocytosis is representing movement of molecule of drug inside the cell, and exocytosis represent outside movement of the cell of the drug molecule from the cell. So these are basic difference between these two terms. So let me clear you the first, endocytosis. Endo means inside, and cytosis is a process of cell which is engulfing any agent, or endocytosis can be of two subtypes. First one is phagocytosis and the another one is pinocytosis. In case of phagocytosis, the first word, the phago, means to eat, to phagy, that is a phagia. That means it is applicable for the solid particle. So if any drug which is in a solid state inside the vesicle can be expressed or termed as a phagocytosis. But if the substance of interest is liquid, then it will be known as a pinocytosis. As its name indicates, pino is meaning of pina, that means Hindi word, that means to drink something, and the drink something is specifically registered or restricted for the liquid one. Coming to their specification, so first point say small droplets are engulfed by the cell. In case of endocytosis, if you are talking about the phagocytosis, so small droplets of the drug molecule will be engulfed or will be taken out by the cell with the help of cell. Cell membrane also support in this process, and they are carried to the cell as a vesicles. So in this case, the drug molecule are turned into vesicles, and that vesicle is bring inside the cell so that the cell will accept that particular drug or accept that particular agent which is being intaken by the cell membrane. Now coming to the second point, vesicle is then broken. The drug molecule or droplet molecule which is converted as a vesicles, this vesicle is now converted or broken down to release the substance which it has taken out. Now coming to the next point, some protein and vitamin B12 are the example of the drug process which is being taken by this endocytosis process. So endocytosis is a specific type of transport of the drug across the cell membrane, and most of the case it is applicable for the molecule which are in higher molecular size. And the example for this endocytosis are protein, because protein are in higher molecular size, along with some kind of vitamin B12 which are taken by the process of endocytosis. Now we have to understand the another term which is exocytosis. So exocytosis means exo, that means outside the cell. So if something is secreted or eliminated by the cell with a specific phenomena, and that is called exocytosis. So what are the points? Reverse process of endocytosis. In endocytosis, suppose this is a cell. So endocytosis, in the process of endocytosis, it is the cell molecule, this is the drug droplet molecule or vesicles molecule. So this is taken up by the cell inside the cell. So this is called endocytosis. But in this case of exocytosis, the cells are expelled outside the body. That is simply example of exocytosis. So what are the specific phenomena of this? So these are, that means exocytosis are responsible for the secretion of neurotransmitter from the nerve ending. In upcoming class, we are going to study about the different types of neurotransmitter, different types of neurohumoral transmission we can say. So all these are based on the process of exocytosis, then where, how and how much quantity of the neurotransmitter will be released from the cell, which, or we can say vesicle, which is stored inside the presynaptic nerve vessel or nerve ending. So the release of neurotransmitter will take place with the help of exocytosis process. So this was theory point of view. Now let me explain you all these theories, summarize all the transport of drug molecule across the cell membrane with the help of diagram. So to take a diagram, to understand the basic concept of the process with the help of diagram, we have to move on the second whiteboard section. So let's move to the next part. Now in this section of whiteboard, I am going to summarize and conclude all the important types of movement of drug across the cell membrane with the help of respective diagram. I am not going to explain this as a theory because I have already explained is in a previous part of whiteboard section. So the first is passive diffusion. Passive diffusion can be understood with the simple diagram. As you can see in this whiteboard section, we have a cell membrane. Cell membrane is made up of phospholipid bilayer. That means membrane having two types of layer. In this diagram, the circle portion representing the head side, and this lining structure is representing for the tail part. So here is a cell membrane. In this cell membrane, allowed the diffusion of drug molecule in two types. For the one, that is a lipid soluble drug, and the other one is water soluble drug. So first let me explain you the water soluble example of movement of drug. In this case of water soluble drug, there should be a aqueous pore in the cell membrane. The water soluble drug can only move through the aqueous pore. Without the help of aqueous pore, they are not able to move across the cell membrane. So this is the water soluble drug. This water soluble are crossing the cell membrane by the aqueous pore, and then after some time they are inside the cytosol of the cell. That is called water soluble drug, can move through aqueous pores only. But in case of lipid soluble drug, there is no need of aqueous pore. They may diffuse across the cell membrane by dissolving themselves in the cell membrane. membrane. So these drugs are lipid soluble drug. As you can see in this diagram, the lipid soluble drug can be easily crossed through the cell membrane, and these two types of diffusion is example of passive diffusion, because in this type, because in case of water soluble as well as in lipid soluble, there is no need of energy. So we can say these are energy independent movement of drug across the cell membrane. Now coming to the second type of movement of drug across the cell membrane, that is facilitated diffusion. I have explained you the all theories. Now compare the theories with the help of diagram. This is another cell membrane, and cell membrane is having different types of movement across themselves. So here these are the drugs. The green circle which I have made here is representing the drug molecule, and this the different types of structure in the cell membrane we can say, or across the cell membrane, are example of drug transporter. Drugs transporter are the agent or movement molecule that can transfer the drug from the extracellular region to the intracellular region, or we can say cytosol of the cell. So the drug came to the drug transporter, they occupy the respective space. You can see the diagram, you can see the angle of movement of drug. Here this side upside is open, so that drug molecule can take part or bind in the drug transporter we can say. After binding of the drug with the respective transporter of the drug molecule, they change their structural relationship. As you can see in this diagram, I have plotted an arrow that represents a bidirectional movement. By this direction they move toward this one, that is drug transporter will change their shape, they change their orientation. Now the drug will release to the intracellular site of region, or we can say cytosol of the cell. So the movement and the changes are taking place respectively, so that the drug transporter can transport the drug from the extracellular region to the cytosol of the cell. Now third one is active transport. Active transport is a transport which require energy to be facilitated. So that is the energy requirement place in this case. As here in the diagram you can see there is also cell membrane, but these are the movement transporter of drug transporter. The drug transporter are transporting the drug with the help of energy. So in this diagram ATP need ADP conversion, then only the movement of drug take place. And the last diagram, that is a endocytosis. We can also understand with the same pinocytosis and the phagocytosis in the same diagram, but endocytosis is a process of engulfing the solid vesicle inside the cell membrane, which are larger in size, so that they cannot go through the other transport mechanism. In this case of diagram, as you can see, these are the large drug molecule, and the large drug molecule are get entrapped inside the cell. Then only cell can use that particular agent for their energy or whatever the reason for that absorption. That means these are the drug, specifically large molecular size of drug, they are engulfed by the cell. This is called endocytosis. But in this case, if the drug is removed outside the cell, that process should be known as a exocytosis. And if the large dose molecule are in solid state, that is called phagocytosis. But if it is in liquid state, that can be termed as a pinocytosis. So with the help of this 4 diagram, we have concluded our topic, which was the movement of drug across the cell membrane. This was an important topic. I hope you have got the point very clearly. So at last, thank you very much for watching this video. The reference for today's whiteboard lecture is KT Tripathi, Padmaja Uday Kumar and P. Jagdish Prasad, and the diagram which you are seeing on the whiteboard I have taken from the Lipin Code. That is another good book. So friend, that's all for today. Thank you very much for watching this video. Please give us your feedback in the comment box. Thank you.